Welcome back. We're going to talk about testicles in this video. So we have a cross-section of a testicle here. The outer layer of the testicle inside the scrotal sac is called the tunica vaginalis. The inner region is known as the tunica albuginea. And then the structures here that are separated into what we call lobules are known as seminal vesicles. And these are where all the real magic of the testicles occurs. So seminiferous tubules here, these are what produce sperm. Within these seminiferous tubules, we have different types of cells. So we have some cells called sustentacular cells or Sertoli cells. These cells nourish developing sperm, and they release a hormone known as inhibin, which decreases sperm production so that we don't have too high of sperm amounts. We also have germ cells, so spermatogonia, that actually produce new sperm, and they're constantly dividing once we hit puberty. Then surrounding the seminiferous tubules, we have these interstitial cells that are also known as Leydig cells, and you can see them over here. These are the cells that produce testosterone. Testosterone is needed for sperm production, but it's also needed to produce secondary sexual characteristics in males. This is just explaining where this picture came from, so you can, be happy with who actually produced the picture, because this is a really cool electron micrograph. So we're going to look at spermatogenesis now. Spermatogenesis is the production of sperm in males. So in your general biology class, you learned about basic gametogenesis when we talked about meiosis. In this class, we're going to focus on male and female meiosis. So we're going to go through actual spermatogenesis, okay? Spermatogenesis begins at puberty when the hypothalamus releases lots of gonadotropic releasing hormone, which stimulates the anterior pituitary gland to secrete follicle stimulating hormone, which stimulates the spermatogonia, and luteinizing hormone, which stimulates the Leydig cells to produce testosterone. So the testes are going to start producing lots of testosterone, which is what is needed to stimulate spermatogenesis. And then, of course, follicle stimulating hormone also is needed to produce or to stimulate spermatogenesis. So normal spermatogenesis takes approximately 2 months to occur, starting with that spermatogonium and ending with sperm. So males are constantly undergoing spermatogenesis. The start times will differ. I mean, obviously, you're not going to activate every single spermatogonium in your seminiferous tubules at the same time, but this occurs continuously. So every single day, you're in the process of sperm production from the time you start puberty, hit puberty, until you die. And so this starts with the spermatogonium here. Spermatogonium will undergo mitosis, producing a primary spermatocyte and another spermatogonium. This primary spermatocyte then undergoes the first meiotic division. And if you remember, in meiosis, we have two major functions that cause each sperm cell to be unique. We have the crossing over in prophase 1, and then independent assortment in metaphase 1. In the end, we have haploid sex cells. These haploid sex cells are then going to go through a second meiotic division because though they're haploid, they're still duplicated. So then we go through a second meiotic division where it's basically very similar to normal mitosis, and we separate the sister chromatids from each other, producing 4 non-identical sperm cells. But these sperms are actually immature. They're known as spermatotids. So spermatids are going to have to go through a process known as spermatogenesis to produce the actual sperm. So I just kind of went through this, but we'll go quickly again. Spermatogonia is a diploid cell that starts the entire process of spermatogenesis. It initially divides via mitosis into a primary spermatocyte and another spermatogonium. So you always have those initial diploid stem cells. The primary spermatocyte is diploid and it undergoes meiosis. It produces a secondary spermatocyte that is haploid, but it is still duplicated. And then we have a spermatid at the end of meiosis 1 that has went through the secondary meiotic division. And so we end up with 4 immature spermatids that are going to now go through spermiogenesis. So spermiogenesis occurs in the following days, where you take that immature sperm, we add an acrosomal cap that contains digestive enzymes to help get into the egg, get through the layers of the egg in the female, the oocyte. We add flagella, which forms onto the midpiece. So this flagella forms, allowing the sperm to be motile. So sperm are immotile initially because they don't have the flagella. They are immotile. They have to have a place to mature, and so they move from the seminiferous tubules into the rete testis. From the rete testis, they move into the epididymis. So sperm move to the head of the epididymis, and they finish spermiogenesis. As they are finishing spermiogenesis, they're moving. So as we're producing more new spermatids, they move into the head, and sperm just keep moving and moving and moving. By the time they reach the tail of the epididymis, they're going to be mature. And so they can be released into the ductus deferens during ejaculation as mature sperm. So what is the ductus deferens, also known as the vas deferens, which a lot of you have probably heard of because most of you have heard of a vasectomy. The ductus deferens is the tube that runs from the epididymis to the urethra. So this is where sperm that are mature are going to enter. They move via muscular contractions towards the ejaculatory duct. So here's that ductus deferens. Here's the ampulla. Here's the seminiferous, or I'm sorry, seminal vesicle. And then here's that ejaculatory duct where both the sperm and the first of the semen are going to enter. The final region of the ductus deferens then is known as the ampulla. So that's the ampulla right here. Sperm move with the semen from the seminal vesicle into the ejaculatory duct, and then they both move into the prostate, or into the urethra. The prostate and the bulbourethral gland are also going to release their own secretions, forming the seminal fluid. So seminal vesicles, which are right here, there's one on each side, obviously, produce both prostaglandins and fructose. Fructose is used to feed the mitochondria of the sperm to allow them to have a good energy supply. The mitochondria are found in the midpiece of the sperm. And prostaglandins help the cervical canal to widen and become easier to move through. So female cervical canal is usually very, very closed off. There's a lot of mucus there, and that mucus is very, very hard and not easy to get through. So prostaglandins help to soften that mucus and widen that canal. The prostate, which is right here, produces mucins that are alkaline to help protect the sperm, and produces a prostate-specific antigen, or PSA, that kind of helps to liquefy the sperm and help it to move even faster towards the egg in the female. And then the bulbourethral gland produces a clear mucus that protects the urethra, but also provides lubrication for sexual intercourse, so that typically when a male is aroused, that there will be liquid at the tip of the penis. That is secretions from the bulbourethral gland. So semen is not just sperm. Semen is formed from the seminal fluids produced from the seminal vesicle, the bulbourethral gland, and the prostate, and sperm. Altogether, it's typically called semen or ejaculate, and it is released during sexual intercourse. Each ejaculate typically has anywhere from 200 to 500 million sperm, and these sperm are going to move into the vaginal canal. By the time they reach the egg in the fallopian tube, there may only be a couple dozen sperm left. So it really depends on how healthy and strong the sperm are. All right, I'm going to stop here. We talked a lot about the testicles and sperm production. We'll get into the penis in the next video, and then we'll start on the female reproductive system. Have a great day. Bye.